You are addicted to something your body was specifically engineered to chase. And that engineering is 15 million years old. Not a metaphor, not a figure of speech. There is a gene sitting in your liver right now, a broken gene, technically a pseudogene, that was deliberately switched off in a European ape during a famine that was killing the entire species. The switch made that ape crave sweet food with an intensity no previous primate had ever felt. It made every gram of sugar that ape consumed get converted to fat at maximum efficiency. And that ape survived. It came back to Africa. It became the ancestor of every human being alive, and the gene never got switched back on. So every time you reach for something sweet without meaning to, that's not weakness. That's not a character flaw. That's a 15 million year old survival instruction running exactly as intended in a world it was never designed to see. Follow the object backwards. Start where you're standing. The fig. Hold it. Small, soft, wrinkled at the stem end, about the size of a thumb. You probably didn't eat one today. Most people don't anymore. But for roughly 26 million years, a fig or something exactly like it was the center of everything. The calories, the water, the sugar, the survival. Before meat, before tubers, before grain, there was fruit. And at the center of fruit was sweetness. And sweetness was the signal your ancestors' nervous systems learned to chase before they had language, before they had tools, before they had anything resembling a plan. Go back. Keep going. 26 million years ago, the African continent looked nothing like it does now. Dense, tropical woodland stretched from coast to interior. The forests were full of apes, not the sparse, struggling populations you think of when you picture early human ancestors, but enormous, thriving communities of fruit-eating primates who spent most of their lives in the canopy, moving from tree to tree, eating ripe figs and wild berries and soft, sugar-heavy pulp off 100 species of plants. The diet was roughly 70 to 80% fruit. The calories were consistent. The sweetness was everywhere. Then, around 15 million years ago, the climate shifted. The planet cooled. Sea levels dropped enough to open a land bridge between Africa and what is now southern Europe. Some of these apes crossed over. They followed the fruit. Northern Africa, the Mediterranean basin, early Europe, it was still warm enough, still green enough, still full of trees that bore sweet things. For a while, it worked. The apes expanded their range, the population grew, and then the fruit ran out. Not all at once. Slowly, seasons got longer, winters got colder. The fruiting cycles that European trees operated on were nothing like the year-round tropical abundance these apes had come from. There were gaps. Weeks, then months, then entire seasons with almost no ripe fruit available. The apes that had crossed into Europe were not equipped for this. Their entire metabolic system, their gut, their brain reward circuitry, their insulin response was calibrated for abundance, not scarcity. They started dying. The fossil record from Europe between 14 and 10 million years ago shows ape species disappearing one after another. The cooling took them species by species until the European ape lineage was almost entirely gone. Almost one population survived. And the reason they survived is one of the most extraordinary accidents in the history of life on Earth. Here's the object again. One fig. Ripe, August, somewhere in what is now southern Europe, roughly 15 million years ago. An ape picks it, puts it in its mouth. And the thing that happens in that ape's liver in response to the fructose in that fig is different from what's happening in every other ape's liver around it. Just slightly. But measurably. That difference is a single gene, or rather, a gene that just stopped working. Uricase, it's an enzyme. Its job in most mammals is to break down uric acid, a waste product produced when fructose is metabolized. When uricase works normally, uric acid gets cleared. Efficiently, cleanly, no buildup. When uricase doesn't work, uric acid accumulates in the blood. An elevated uric acid, researchers at the University of Colorado Anschutz discovered, does something remarkable. It amplifies the fructose-to-fat conversion pathway. It makes the body store calories from sweet food far more aggressively than it otherwise would. It essentially turns a single piece of ripe fruit into a more powerful caloric investment. Now, in a European summer with a limited fruiting window, That is not a disadvantage. It is the entire difference between living and dying. The ape with the broken Uricase gene didn't need to eat more fruit than the others to store more fat. It ate the same amount. And got fatter. Got through the winter. Had offspring. Those offspring inherited the broken gene. They got through their winters too. The Richard Johnson Research Group at the University of Colorado, working with colleagues including Peter Andrews from University College London, published their analysis of this exact mechanism in Scientific American in October 2015. They found that all living great apes, orangutans, gorillas, chimpanzees, bonobos, and every human being on Earth carries a non-functional pseudogene where uricase used to be, not a different version of the gene, a broken copy. A gene that evolution deliberately kept non-functional because the populations that lost it survived, and the populations that didn't, the European apes, are gone. If that just connected something in your head, hit like. It literally determines how many people see this. Now track forward, because the gene was only half the mechanism. The broken uricase gene made fructose storage more efficient, but that only mattered if the ape was motivated to find fructose in the first place. Which brings you to the other side of the system. The hunger side. The craving side. The part that is sitting in your brain right now, running in your tongue, there are clusters of cells sitting just beneath the surface. Taste buds. Inside each one, specific subtypes of cells produce different receptor proteins. One subtype produces what is called TSAS1R2, TSAS1R3, the sweet taste receptor. When sugar molecules contact this receptor, it fires a neural signal that travels up to the brain. The brain registers the signal and does two things simultaneously. It releases dopamine, and it tells you to keep eating. Dopamine is the molecule of motivation. Not pleasure, motivation, the distinction matters. A 2021 study in the journal Frontiers in Psychiatry, reviewing decades of animal and human data, found that dopamine release from sweet taste is specifically driving the urge to seek more, not just the enjoyment of the thing you already have. You don't just feel good when you taste sugar. Your brain immediately begins organizing your behavior around getting more of it. And here is the part nobody taught you in school. This receptor, TAS1R2/TAS1R3, is not a recent arrival. The genes encoding it are found in fish, in lizards, in rodents, in pandas, in bats, in every primate that has ever been sequenced. They have been in place, conserved by natural selection for hundreds of millions of years, because every animal that had them survived better than the ones that didn't. The craving for sweetness is not a human quirk. It is one of the oldest survival instructions in vertebrate biology, which means the species we find most interesting for comparison are not the ones who have the gene, they're the ones who lost it. Cats, domestic cats, house cats, tigers, lions, obligate carnivores across the board. In 2005, researchers published in PNAS a finding that stopped the field cold. The TES1R2 gene in cats is a pseudogene, broken, non-functional. Cats cannot taste sweetness at all. Put a bowl of sugar water in front of a cat and they will walk past it without interest. Because for an animal that eats nothing but meat, the ability to detect fruit sugar is useless. And natural selection, operating on the use it or lose it principle, switched it off, probably 40 million years ago. So here are two gene switches running in parallel. In meat-eating cats, the sweet taste gene got turned off because sugar wasn't worth chasing. In European apes, the uricase gene got turned off because storing more sugar was worth everything. Both changes were adaptive, both were permanent. One is why your cat doesn't care about your candy, the other is why you do. Hold on, because here is the stop on the journey that changes what everything before it means. The Hadza people of northern Tanzania are one of the last remaining hunter-gatherer populations on Earth. Anthropologists from Arizona State University and University College London have been studying their diet systematically since the 1980s. What they found about honey stopped researchers cold. During rainy season months, when the acacia and baobab trees flower and wild bee colonies peak, honey accounts for between 15 and 20% of total Hadza caloric intake, primarily among men. Not a taste, not a treat, a staple, and they don't collect it passively. Hadza honey hunting is a full-body, hours-long commitment. You find a hive, often 30, 40 feet up a tree. You build a fire at the base, let the smoke sedate the bees, and climb. You smoke your hands. You reach into the hive and pull out dripping honeycomb with your bare hands while bees are stinging your arms and face. You do this because the caloric payoff, honey is between 80 and 95% sugar. The highest caloric density of any natural food is worth exactly that level of pain and risk. And this is where the tour guide gets quiet for a second, because you have to picture the bird. Greater Honeyguide. Indicator, indicator. Latin name chosen apparently by scientists with a dry sense of humor. A small, drab-looking bird that lives throughout sub-Saharan Africa and has one very specific behavior. It finds wild bee colonies, then finds humans and leads them to the hive. It calls. It flies a few meters toward the hive. It waits. It calls again. When the humans follow and the hive is opened, the bird eats the wax and larvae left behind. The humans get the honey. A 2023 study published in Science by Cambridge researcher Claire Spottiswoode found that Hadza honey hunters and Yao honey hunters in Mozambique each use distinct culturally specific calls to recruit honey guides. And the birds have learned to respond preferentially to the calls of their local human partners. The cooperation between humans and honey guides is believed to be ancient. Some researchers estimate it is tens of thousands of years old. We do not know exactly when it started. The honest answer is that we can't be sure. But rock art depicting humans climbing to reach beehives, with birds visible nearby in some cases, has been found across Africa, Spain, and India, some of it dated to between 8,000 and 40,000 years ago. The craving for sweetness was so strong that it drove humans to build a cross-species working relationship with a wild bird. That is not hyperbole. That is just the archaeological record. Now go one more stop forward, because here is where the journey turns. The Uricase mutation, the sweet taste receptor, the Honeyguide partnership, the fig-centered ecology, all of it was operating in a world where concentrated sweetness was rare. Seasonal, earned, a famine survivor mechanism in a world that still ran on scarcity. A hunter-gatherer in pre-agricultural East Africa, depending on who you ask. Probably consumed somewhere between 2 and 4 teaspoons of added sugar per day, almost entirely from honey and ripe fruit, and only when available. The American Heart Association's current guideline is a maximum of 6 teaspoons per day for women and 9 for men. The average American consumes 17 teaspoons of added sugar per day. The mechanism built for scarcity is running in abundance. The broken uricase gene that helped one European ape survive a fruiting gap 15 million years ago is now driving fat storage in a body that never experiences a fruiting gap. The dopamine reward system that once fired when a Hadza man pulled honeycomb out of a tree now fires when someone opens a bag of processed food in a gas station at 11 p.m. The system did not change. The food environment around it changed completely. In roughly 10,000 years, a geological eye blink, we went from a diet where concentrated sweetness was rare and hard to obtain to a diet where it is the cheapest, most available thing on the shelf. And the body has no mechanism to compensate. There is no satiety signal for sweet the way there is for fat or protein. In a 2011 paper in Frontiers in Systems Neuroscience, Ivan de Araujo at the John B. Pierce Laboratory in Connecticut found that the dopamine response to sweetness does not fully habituate. It doesn't get bored. You can eat sugar for an hour and the neural signal doesn't say enough. We've handled this. It keeps motivating more intake. That is not a design flaw. In an environment where sweet food appeared twice a year, that response was exactly right. Eat everything you find as fast as you can because it won't be here tomorrow. Subscribe if you want the follow-up, because what happens to this system under modern food engineering is the part that genuinely should change what you buy at the supermarket. Go back to the object one last time. The fig. You probably didn't eat one today, but the ape that did 15 million years ago in a cooling European forest, in a body that had just lost a gene it would never get back. That ape was running a survival system so well calibrated to its world that it outlasted every other ape in Europe. And then it walked back to Africa, and it became everything. The craving you feel isn't the problem. It never was. It was the solution. The most elegant, metabolically precise, behaviorally powerful survival tool any primate ever developed. The craving kept the line alive through ice and famine and 15 million years of uncertainty. The world just changed faster than the gene could keep up. And now, the thing that saved the species is charging it a different kind of price. That's not a tragedy. It's just where the journey landed. Tonight, what surprised you most? The broken gene, the Honeyguide bird, or the fact that the cat sitting near you right now literally cannot taste what you're eating? Drop it below. I read them.